Welcome back to our series of lectures on neuroendocrine regulation 2, cellular neuroendocrinology. This is the second in a series of 2 on chapter 7, and then subsequently there'll be a brief recording on chapter 8. So we continue with chapter 7 in our 7-2 lecture here, regulation of hormone synthesis, storage, release, transport, deactivation. So we're going through these sections, 7.2 to 7.6 in the textbook, and we'll follow that series. This I like to use to discuss, I'm going to set my pointer, excuse me here, the synthesis of peptide hormones. This renin-angiotensin system is classically taught in physiology courses as a peripheral multi-organ endocrine system, and we'll explain that in a moment. But this entire system is sorry about that. This entire system is contained within certain neurons within the brain as well. I just like the complexity of the processing and how well it's known to help make some points about post-translational processing and pre-pro hormones and things of that sort. So typically the angiotensinogen is produced by the liver. It would be pre-pro, and then this is the pro that would be secreted into the plasma. And then the kidney is the source of the renin that converts angiotensinogen to angiotensin 1. It's a peptidase, a cleavage enzyme. There are drugs that can block there at this step. And then the angiotensin 2 circulates in the blood, and angiotensin converting enzyme, or ACE, is located primarily on the surface of endothelial cells in different vascular beds. One of the highest vascular bed concentrations is in the pulmonary tree, pulmonary vascular tree. But it's distributed in other areas as well, such as in the kidney, where the conversion of angiotensin 1 to angiotensin 2, I don't know why this keeps doing this to me. I'm just going to, oh, there we go. Sorry about that. And a new version of Windows. The angiotensin 2 can bind to receptors locally in the kidney or distributed throughout the vascular bed. And there are two types, angiotensin type 1 receptors and the type 2 receptors. And there are drugs that can inhibit, like ACE inhibitors here, that'll inhibit ACE, but there's other enzymes. Remember, all peptides can be acted on, deactivated, as we're continuing through this idea of going from active to inactive by peptidases. So there are non-ACE chimases and these kinins that can also, in kinase 1 as an example here, or maybe that's 2, sorry, that can be involved as well. So it's not just the ACE that is active here, there are others. And then of course angiotensin 2 is going to be continually broken down, angiotensin 3, angiotensin 4. You can look in the literature if you want to follow up on that, until ultimately there's a biologically inactive form of this. I think this is an octapeptide, so it's only 8 peptides to begin with. So a couple more cleavages and you're down to inactive dipeptides and single amino acids. The angiotensin 1 receptor is responsible for vasoconstriction, heart contractility, aldosterone release, although I really don't think that this one, I think it's the AT2, but I'd have to look that up. Glomerular filtration, renal blood flow, vascular hypertrophy, and left ventricular hypertrophy, and heart disease. And then the inhibition of cell proliferation, possible role in growth and differentiation for the AT2. And as I said, I double check, I think it's responsible for aldosterone release in the adrenal cortex. So this is a nice way to illustrate from the prohormone to the most biologically active form additional cleavages to two other angiotensin 3 and then angiotensin 4 as it's continued to be broken down, biologically active forms, and the biological effects of each of these vary slightly, not only based upon receptor subtype and localization, but on the nature of the binding peptides themselves. So essentially this model system can be applied to any peptide hormone, and all these events will occur in different locations. So here in this, my favorite illustration of the anterior pituitary gland, we try to picture all of that occurring within here up to the point of secretion at number 7. So let's get oriented first. We have a capillary with red blood cell in it. So this is the plasma fluid compartment here. And this is the anterior pituitary. So we're picturing hypothalamic releasing and inhibiting hormones passing through the fenestra, diffusing down their concentration gradient from the blood to the interstitial fluid and bouncing around out here until they find their receptor. This is a lactotrope, and this is a somatotrope here for growth hormone, and there's another lactotrope over there, but somewhere else this might be a gonadotrope of FSH or LH, whatever. And these follicular cells that are a bit different, and you can read up on that if you want. And of course the fibroblasts to maintain the extracellular matrix. Fibroblasts in the extracellular matrix are the stroma, and then these functional cells are the parenchyma of this, and then of course here's the nucleus of the mitochondria of the endothelial cell. Okay, so we have our orientation. The ligand is going to bind to the receptor on the membrane, the surface membrane of the target cell, and it's going to impact synthesis and secretion. So DNA and RNA to the rough ER, transport vesicles to the Golgi, packaging in the Golgi, producing the secretory vesicles. In this case, in the pituitary, multiple vesicles fuse together to form these large secretory granules. And 6A here, of course, is the internal mechanism for the destruction. It might be autophagy, and typically we envision this as a lysosome, that would destroy the contents, break it down, and recycle the amino acids, right? And then here to the 6, the process of bringing the secretory granule from some position subsequent in the cytosol, subsequent to the Golgi, to the plasma membrane, where a local event can bring it all the way to dock and fuse and then open up for the process of exocytosis, is what we want to focus on. How does this occur, and what is it dependent upon in general, right? So synthesis and secretion are what we're considering here. And there might be post-translational processing that might start, from pre to pro-hormone, and then pro-hormone cleavage can occur anywhere along this route, and the biologically active form of the molecule would be secreted. In the case of the hepatocyte, with the angiotensinogen, it would be secreting the pro-hormone, right? So this can be different for each type of hormone system. Now, what about for the steroid synthesis? So let's go back for a moment. They're not showing smooth ER, but there would be smooth ER, and we'll see more of that as we proceed. Smooth ER is where the lipid synthesis occurs, to be generalizing about it. And these, of course, are the steroid hormones. So where are they synthesized? Or at the smooth ER. And they depend upon cholesterol as the ultimate precursor substrate, and can be converted to pregnenolone, and then around the progesterone, or to the progesterone, and from the progesterone to the aldosterone, corticosterone, or to from the 17-alpha hydroprogesterone to the cortisol or hydrocortisone. And then, of course, the 17-alpha progesterone to the androstenedione, androsterone to testosterone, and you see the locations where these occur, of course, and then the 17-beta estradiol. So these enzymes for the synthesis at each of these arrows, and there probably are more arrows, but anyway, this represents some of the major steps. Those are translated at the ER and then moved over to the smooth ER, and we'll see that a little bit later. These molecules are lipid soluble, so as soon as they are made, they're diffusing away. The cholesterol is transported in, these are being produced, and they are diffusing away down their concentration gradient out into the interstitial fluid and then to the plasma. Along the way, if they encounter another enzyme, they'll get and in the pathway they'll get converted to something else. So it's this game of moving away and getting converted to something else and something else. And so the concentration of enzymes in this localized area has to be high enough so that you can't, not too much of the androstenedione escapes the cell into the blood without encountering the enzyme necessary to take it closer to testosterone. But many of these intermediaries can be detected in the plasma and the urine, and certainly in the tissue that's synthesizing it. So this is the way to think about the difference of the synthesis and secretion of steroids versus synthesis and secretion of the peptide and protein hormones. Well, now let's think about the release or the packaging of the non-steroid hormones, right? Peptides and proteins. So we'll compare and contrast the function of synaptic vesicles and secretory granules. And I'll just read this to you so you don't have to read it. The way by which secretory granules acquire their contents is different in vesicles and granules. Packaging into synaptic vesicles is performed from cytoplasmic pools. Hence, synaptic vesicle membranes contain 1. Electrogenic pumps making the inside positive with respect to the outside, and 2. Chloride channels which dissipate the membrane potential and allow the buildup of an acidic environment inside the vesicle. After releasing their contents, the secretory vesicles reform. They pull back away from the membrane into the cytosol and fill with transmitter from the cytoplasmic pools. The electrochemical gradient generated by the proton pump drives the accumulation of transmitters via several classes of neurotransmitter transporters. So the VMAT, vesicular monoamine transport, is one of the examples of that, and I think there's different isoforms of that, VMAT 1 and 2, maybe something of that sort. You can check on that if you're interested in it. On the other hand, since secretory granules contain proteins or polypeptides, they cannot refill in the cytoplasm after undergoing exocytosis. So the membrane has to be inserted, the vesicular membrane or the secretory granule membrane is inserted into the plasma membrane, and then it has to be recycled, and we'll take a look at that in a moment. So the packaging into secretory granules occurs in the trans-Golgi network, and we'll explain that. We know it starts at the rough ER. So I like this generic illustration of the cell. It shows us what we need, reminds us that there are housekeeping proteins like the cytoskeleton and all the enzymes out here for the glycolytic pathway, and proteins in the membrane for transporting glucose in, of course, and things of that nature, just to keep the cell alive. And here's the centrioles and things of that nature. So we're not thinking so much about the housekeeping genes and the housekeeping proteins or gene products. For this illustration, we're picturing the gene of a protein hormone being expressed in the nucleus, and the mature mRNA, after the RNA processing, splicing, and poly A tail and 5' cap, then can pass through the nuclear pores into the cytosol. If the mRNA has a signal peptide sequence on it, then it'll associate with ribosomes that will then translocate to the ER. As they begin to produce the peptides, they'll then become attached, and translation will continue, as we'll see in just a moment, not going into any level of detail. And then we know that the movement of the proteins will bring it to the Golgi and then to the vesicles for the fusion. This is the classic omega figure. What I like about this is it reminds us a little bit of the three-dimensional aspects of all this. We always see these as little circles or two-dimensional things, but they're spherical properties are a little more well illustrated here in this 3D image. Note that the rough ER and the smooth ER are continuous, so the enzymes may translated here at the rough ER for the synthesis of the steroids. Some of it occurs at the mitochondria, and then some at the smooth ER, but anyway, we'll just bypass that. But lipids in general for the cell, the phospholipids for the phospholipid bilayers of the membranes, are produced at the smooth ER. So some of those are membrane enzymes, synthesizing enzymes are membrane proteins, and they'll just be shuttled through the membrane out here to the smooth ER. Or if they're in the lumen, they'll just be shuttled within the lumen from rough ER into smooth ER, and then they'll perform their job there until their life is over, right? And they get degraded just like any molecule. Here we're talking about synthesis, secretion, and the sustaining biologically active levels, and then degradation. For hormones, that's the life cycle of any biological macromolecule, right? It has to be made, activated, do its job, and then be inactivated either for degradation or to an inactive state where it can be recycled to an active state. So always keep that in mind when we're talking about these things. And this is how we generate a plasma level of a hormone, right? If this is a corticotrope, then ACTH is being pumped into the blood and then degraded after it's been in the blood for a period of time, and the synthesis and secretion of the collective corticotropes in the anterior pituitary and all the processes in the blood to eliminate, set up a clearance rate for the ACTH establishes the plasma levels of ACTH at any one point in time. So this is the way we're trying to think about this. So let's go through this Bio 1 illustration and see a little bit more of what's happening here. Here we've got the little green protein to kind of help us picture what's happening. So picture this little green protein moving over into the smooth ER, or somehow to the mitochondria, and the mitochondria near the smooth ER collectively working together to generate the steroids that we've talked about. So a steroid synthesizing cell in the adrenal cortex or the gonad will have a large amount of smooth ER in it. Anterior pituitary cells producing peptide hormones are going to be filled with rough ER. There should be no cell that has none of either of these, because we need to make our phospholipids for our membranes here, and membranes don't last forever, even though there's a recycling, as we'll see here. Lipids get old and you got to get rid of them too. So that's the way to think about it. So the DNA instructions for building polypeptide chains leave the nucleus and enter the cytoplasm. Well, that's the mRNA, as we had stated before, through the nuclear pores. The chains, green, are assembled on ribosomes in the cytoplasm. Okay, we're not seeing the free ribosomes here for housekeeping proteins, but we're seeing the proteins as they're being produced and inserted into the ER. This may not be totally accurate. I don't think there's too much in the cytosol as this is being produced. It's all happening right at the ER membrane, and then the protein only goes into the lumen or is immediately inserted into the membrane of the ER if it's a membrane-bound protein. Either way, shuttled ultimately to the Golgi. So the secretory pathway, some polypeptide chains enter the space into the inside rough ER. Modifications begin that will shape them to the final protein form, post-translational processing. In the membrane of the smooth ER, lipids are assembled from building blocks delivered earlier. Vesicles bud from the ER membrane and then transport unfinished proteins and lipids to a Golgi body. And then the proteins and lipids take on final form in the space inside the Golgi body. Different modifications allow them to be sorted out and shipped to their proper destinations. And then the vesicles budding from the Golgi membrane transport finished products to the plasma membrane. The products are released by exocytosis. So we're going to get into some detail of this related to neuroendocrinology. So some vesicles form at the plasma membrane and then move into the cytoplasm. These endocytotic or endocytic vesicles might fuse with other membrane with the membrane of other organelles or remain intact as storage vesicles. So you're putting membrane in, you're taking membrane out, and then this membrane can be delivered to various targets. It's quite a complex system, the recycling of the membrane. Some of them may go to lysosomes, as you know. It all depends on what is to be done with the contents. That's the content of the membrane itself, membrane proteins and lipids, and within the lumen of the endocytic vesicle. So different destinations for those. And there's different types of vesicles. We're looking at the clathrin-coated endocytotic vesicle here. And as well, things are happening at the basolateral membrane for secretion and endocytosis as well. So we need to keep these basics in mind as we're thinking about the specifics that we're going to be considering in the next few slides. Exocytosis is of two types, basically: constitutive secretion. So for the fibroblast we saw in the illustration of the pituitary, the anterior pituitary, the collagen would be constitutively secreted. It might be regulated a bit up and down, or modulated, let's say, but in general it's just going to be put out at a rate that should equal the breakdown in the interstitial compartment, such that you maintain the proper amount of collagen all the time. So there's an unregulated membrane fusion for this, so there might be changes in the total synthesis rate, the expression of the gene, translation, and so forth. So you may still have a constant movement of membrane, just the number of these blue lines inside could be changed based upon synthesis, right? So this would be the case of albumin in the liver, so that or maybe some of the transport proteins that we'll see, but albumin is one of those. If someone has a very poor protein diet, the plasma albumin levels will decrease. Simply, there's not enough amino acids to go around, so there are fewer blue lines of albumin, but still maybe the process. I mean, if your nutrition isn't good, maybe you don't have the ATP that would be involved in some of these steps if it is ATP dependent. Anyway, if this was a constant rate, the delivery trucks on the highway or the trains on the railroad tracks, or the ships in the ocean, anyway, if they're moving at the same rate but they're carrying less, then less is delivered, right? So this is what we mean. It's maybe an unregulated membrane fusion, but the newly synthesized plasma membrane protein, or the soluble proteins for constitutive secretion, those can be regulated. Okay, so that's a constitutive secretion, and then what we're thinking about, of course, is a ligand binding to a receptor with signal transduction that will trigger the movement of the mature secretory vesicle or secretory granule to the membrane, causing docking, fusion, and then the exocytosis for regulated secretion. So let's take a moment to look at this brief review article from the European Journal of Endocrinology, 1997. Exocytosis in excitable cells is conserved molecular machinery from yeast to neuron. So again, we're moving from an introductory textbook to review literature to kind of catch up, or just to get a sense of the transition from an introductory textbook to review literature to see how things get updated. I know this is old by the time you're seeing it, but still it represents that same transition. So your challenge would be to go to find a more current review article on this matter and see what knowledge has been gained between 1997 and the year in which you're studying this material. But essentially, this is the situation. So the blue is the transport vesicle, and this is a membrane protein that we see on it. This is the target membrane, presynaptic grid if it's the neuroendocrine cell, and there's a protein on it. So there's a vesicular snare and a target snare, the V snare and T snare. So the snare hypothesis is that each transport vesicle is proposed to be endowed with one or more unique V snares, generally related to synaptobrevin. So there are many different types of these. And they pair with the cognate T snares, generally related to syntaxin and or SNAP25, thereby docking the vesicle to the correct target. The general fusion machinery assembles on this scaffold, shown in B. So we see the SNAPs and the NSF. It's ATP dependent, remember, like I was saying. So if you're undernourished and you can't make enough ATP, that rate of fusion, even if it is constitutive, is going to slow. So the ATP will contribute to the removal of the SNAPs and NSF, and then that brings about the docking and the fusion of the membranes. So there's a few more steps in here, but then you see the snares dissociate from the vesicle. Let's see. So the fusion machinery consists of cytoplasmic proteins that are readily available to all cell membranes, including ATPase NSF and the SNAP proteins. SNAPs, of which there are at least 3 subtypes, bind to the snare, SNAP receptor complex, enabling NSF to bind. ATP hydrolysis by NSF is essential for fusion, leading to bilayer fusion. Whether additional proteins are required for complete fusion is not yet known. So there are some other properties of the proteins associated with this complex here, even beforehand represented in this arrow here, that we want to think about. We're not going to get into the details. This is enough. SNAPs, NSFs, and snares, and the ATP dependency is sufficient for the critical thinking level integration that we want to have in endocrinology. So what property causes the movement of the contents of the vesicle into the interstitial fluid? Obviously, it's concentration gradient, right? They're diffused down the concentration gradient. Now, there might be some propulsion if, say, this is a secretory granule. If this omega figure totally flattens out, right? So picture this like pushing in, then it's going to dump the contents, right? If there's a complete insertion. But we said the synaptic vesicles will do a kiss and run is one of the hypotheses. So at this point you get the diffusion, and then this will close off, and then the vesicle will come back off, most likely assisted by other proteins. And remember, any movement, we think of the cytoskeleton being involved. Cytoskeleton, the general definition, cell shape, and any movement within the cell. So keep that in mind. But basically, movement of hormones is concentration gradient, from high concentration to low concentration gradient. You always have to ask, how is the concentration gradient maintained? So ideas on release. The adenohypophysis is another endocrine tissue composed of different cells secreting a variety of hormones. So we've seen our illustration on that. These are stored within the cells in dense core granules and are released by appropriate stimuli. So we're talking about the regulated release model. In addition to dense core granules, anterior pituitary cells contain small translucent vesicles, which are very similar to synaptic vesicles, not only in their morphological appearance, but also with regard to their protein constituents. For example, chromogranins and secretogranins found in secretory granules and small vesicles of endocrine cells have also been detected in their neuronal counterparts. So what might these granins be doing? That's not a focus of our conversation today. It's a question for you, if it interests you, to begin to investigate it. So it is also noteworthy that membrane proteins from synaptic vesicles, such as synaptotagmin, a calcium sensor protein, okay, right? So here we go. We have a divalent cation. Most of you should already know that calcium is required for exocytosis. This is what we're building up to, of course. What evidence substantiates that for neuroendocrinology? These have been found associated with secretory granule membranes. Okay, so I said there's additional proteins that might be involved. that are sensors, and they're sensors for calcium. So how might we collect data to substantiate or support this hypothesis of a calcium dependency? So I use this article, 1999. Again, it might be dated, but it might be a landmark one as well. So pulsed laser imaging of calcium-21 influx in a neuroendocrine terminal. So they use the posterior pituitary, where the terminals are really very large, and so they're quite easy to harvest, and they can be visualized, suspended in solution and studied and visualized with proper types of tracers. In this case, it is a calcium sensitive tracer that will generate fluorescent signal. And what they are doing is converting the intensity of that fluorescent signal based upon a ratio, right? So this is their scale bar. If it's higher than if it's equal to the extracellular, it's 1. If it's higher, then it's up to 2, right? It goes to the red. So red is more calcium, is the way to think about it. So here in panel A, they used a pulse laser calcium imaging of voltage activated calcium influx. So in A, this trace, notice time in milliseconds and current. So a downward deflection is an inward current. Okay. If a current is a movement of a charged particle, so if calcium moves in, there should be an inward deflection of this current, a downward deflection. So an influx, right? The calcium influx. So A is a trace showing currents evoked in a patch clamped terminal. So these are big terminals. They can isolate them and see them and clamp them by a 30 millisecond step from a holding potential of -80, typically resting, to 0, definitely going to cross threshold, in the presence of potassium and sodium channel blockers. So they don't want any endogenous depolarization. They want to control the depolarization by their patch clamping, so they block the sodium and potassium channels. Make sense? Excuse me. In addition, the only current, when they're measuring the positive current, will come from the calcium divalent, and they can image the calcium so they know it's moving. Anyway, the terminal was illuminated with a pulse laser light at the arrow up here, right? And that's when they captured these images. To excite the calcium indicator rod 2, 300 micromolar is what they used of that. The resultant fluorescence image depolarization was compared, so that's this one here, you see the red in it, was compared with that obtained by a laser pulse triggered 1 second earlier, the control up here on the left. The ratio of the two images indicates the spatial distribution of calcium elevation. Light measured outside of the terminal has been digitally subtracted to take it to the blue level. Scale bar in this and subsequent figures is 5 micrometers. See, so these are pretty big terminals. Okay, and then in panel B, a series of binary images of the same ratio image created by using the indicated threshold values. And we're just going up through, and we see as you change these threshold values, then you get more and more calcium coming in. And what they can do over here on the right is they do a standard curve, right? So the F to F min normalized 1, 2, 3, 4, 5, and they know the calcium concentration, right? So this is kind of a standard curve, and then that lets them determine how much calcium, what the calcium concentration may have been in any one of these images. Okay, so this is a large balled up terminal, right? It may not be the actual terminal size. When they fragment these, it might be the terminal itself and some of the axon proximal to it, and when it gets torn off, it closes up and then just turns into this big basketball, right? But they start off being big, so that helps. Now we see in this figure 3 our trace again, time in milliseconds, so it's longer, 16 milliseconds, and the currents in nanoamps, and we see an inwardly directed current over here, so that's the calcium influx. And what they did is that's a tail current in the presence of EGTA. So EGTA is a calcium chelator. So if they clog up chelate the calcium extracellularly, it can't get in, right? And they do a dose response. So 0 millimolar, 1.1 millimolar EGTA, and 1 millimolar, and then 5 milliseconds, 10 milliseconds, 20, and 30 milliseconds. And so they're just showing the progression of the calcium influx. Okay, so the trace shows currents evoked in a terminal by stepping from a holding potential of minus 80, remember, resting membrane, or very resting, hyperpolarized maybe, to plus 40, so they're swinging higher this time, basically up to the level of an action potential for 10 milliseconds. So little or no current was evoked during the step, but a large inward current followed it. So a fluorescent image obtained at the end of the step showed little or no increase, whereas 0.6 or 2 milliseconds later showed distinct submembranous rings of increased calcium. Note that there is no evidence for localized sites of calcium influx. So there was a hypothesis of the clustering of these voltage-gated calcium channels, and the evidence here is that it's more diffusely distributed. And again, like I told you, we don't know how much of the proximal axon got incorporated as it snapped off and became part of this basketball, and then would the calcium voltage-gated calcium channels redistribute? Right? So there are arguments to question it, but this evidence seems to indicate there isn't clustering. Okay, endogenous and exogenous buffers compete with the calcium, therefore limit the apparent diffusion of calcium away from the influx sites. Right? So we're not seeing the diffusion because the calcium gets bound by the fluorescent marker. In the presence of added EGTA, the rings of elevated calcium were smaller and had lower intensity. Pattern of calcium influx, however, remained homogeneous, supporting the conclusions that calcium channels are not clustered in large numbers. So those are nerve terminals in the posterior pituitary and should reflect the anterior pituitary. We'd see the same thing if we were looking at a pituitary cell, in this case a lactotrope. And so the ligand is going to come and bind to its receptor. I don't like that this is a lactotrope because there's no stimulating hormone. This is actually constitutive, and dopamine binds to a receptor and inhibits the process. So the lactotrope isn't the greatest example, so let's pretend it's one that depends upon its ligand, say GnRH binding to its receptor. And so what's going to happen proximally here to bring about the calcium change? That's the question we're going to ask. So we know that virtually all the releasing hormone receptors on the target cells in the anterior pituitary are coupled to adenylate cyclase. And so we can see cyclic AMP being made in here and diffusing away from, there's receptors all over the cell, and the GnRH is going to diffuse. There's other capillaries spread around over here, so it gets to all surfaces of this three-dimensional, we're now calling it a gonadotrope, okay? Or growth hormone releasing hormone up here in the somatotrope, right? So we're picturing the cyclic AMP diffusing out. What does it do? How does it affect synthesis and secretion? Right? How do we get a coupling of those two? So what about the cyclic AMP? Measurements of cell membrane capacitance. Now you need to remember what a capacitor is. It is a set of plates, phospholipid bilayer, separated by about 8 to 10 nanometers. So a capacitor is two plates separated by some distance, and of course, connected to a battery source. This is the charged ions outside and inside. And they collect, concentrate, and separate oppositely charged, opposite charges. Let's just put it that way. Ions or ionic constituents. They could be proteins or just electrolytes, right? In our case, we're thinking of calcium. So if calcium gets on the inside and is collected near the inside of the membrane, then it's going to collect positive charges on the inside of the membrane where it's usually negative, right? So membrane capacitance is what we want to keep in mind. If it changes, then there's a change, and it's applied to the investigation of intracellular calcium concentration on exocytosis of neurons and neuroendocrine cells. So intracellular calcium concentration can be modified by either dialyzing the cytosol with different calcium-containing pipette solutions. So you poke your pipette into the cell, and then you dialyze calcium into it, or changing the membrane potential with through some mechanism like patch clamping, as we saw before, to force the voltage-gated calcium channels to open. This last one will tell whether it's dependent upon the voltage-gated calcium channels. Is there another source of calcium inside the cell that can contribute to a transient change in the cytosolic free calcium levels? Yes, the ER, right? The endosomal system. So we're thinking of the IP3 receptor and IP3 second messenger system. Okay, so high calcium resulted in a large membrane capacitance increase, demonstrating the role of intracellular calcium concentration in the stimulus secretion coupling as previously suggested in pituitary cells. Interestingly, in some pituitary cells, such as lactotropes, a tonic calcium influx through voltage-gated calcium channels measured at resting membrane potentials was found to play a major role in secretory activity monitored by membrane capacitance. So the lactotropes have, remember I said that there's a constitutive secretion of prolactin, and it depends upon the dopamine from the hypothalamus to inhibit it, right, to hold it back. So a prolactinoma can be effectively treated with D1 agonists, dopamine D1 agonists. Okay. So a number of other signaling pathways have also been suggested to modulate normal release from pituitary cells. In gonadotropes, breakdown of phosphodienocytides induces calcium oscillations which trigger rhythmic exocytosis. Excuse me. So this is important to understand oscillations of calcium waves and the rhythmic exocytosis. We'll start looking at some of the properties of the hormones 2 in the plasma in a moment. Right, so think back on this of how is the exocytosis triggered. If it's through calcium oscillations, you get rhythmic exocytosis. What are the short-term plasma levels going to look like? So in lactotropes, cyclic AMP was found to increase the magnitude and rate of calcium-induced exocytosis. So this is based on the idea that there might be a prolactin-releasing hormone that can take it above baseline. Remember I said even constitutively released can be modulated, right? So here we're thinking of the dopamine is really like regulating, putting the brake on the system, and then there may be a gas pedal that can modulate it higher, but it's based upon a constitutive rate. So if for some reason the dopamine is blocked, the D1 receptor antagonist, then prolactin levels go way up. So in contrast, cyclic AMP had no detectable effect on membrane capacitance when intracellular calcium was low. So somehow the cyclic AMP is just modulating the calcium situation when the calcium is level. So when the calcium is present, in the cytosol. Okay, so just little insights. I don't give complete stories to keep you thinking about it and wondering and maybe turning to the current literature to get more information. So we now are thinking of how does cyclic AMP function? What role does it play? Well, let's look at some of the diverse effects that it can have in different target tissues for different hormones, chemical messengers, and again see their effect. So in adipose tissue, epinephrine, ACTH, and glucagon will increase triglyceride hydrolysis and decrease amino acid uptake. Right, so favoring the burning of fat. Right, so these help you get skinny. help you stay alive by turning to your fat reserves for energy. Let's put it that way, right? The liver is a target organ. Epinephrine, norepinephrine, and glucagon increase in conversion of glycogen to glucose, so glycogenolysis. Inhibition of synthesis of glycogen, so decrease glycogenesis. Increase glycogenolysis and decrease glycogenesis. Increase in amino acid uptake. The amino acids are then deaminated and used for gluconeogenesis, so an increase in gluconeogenesis. So again, at times of low caloric intake, right, where blood glucose levels will be dropping, glucagon is going to go up. You want to make glucose in the liver and get it into the blood. In cases of stress, and strenuous exertion, right? They kind of go hand in hand. A dinosaur chasing you, you're running for your life. It's a strenuous exertion. So is a marathon, right? So anyway, these all fit into that survival situation. Then we switch to so survive and then reproduce. So here's our reproduction. Target tissue, the follicle, FSH and LH, increase in the synthesis of estrogen and progesterone. So the cyclic AMP is going to impact the activity of the biosynthetic enzymes themselves and their expression, right? Synthesis and activity in this case, and the activity of the biosynthetic enzymes for these lipid-soluble molecules is directly associated with the rate of release. The faster they're made, then the faster the plasma concentrations will increase. The adrenal cortex, ACTH, will increase in the synthesis of aldosterone and cortisol. In cardiac muscle, epinephrine will increase the contraction rate. The thyroid, TSH, will increase the secretion of thyroxine, and that's quite a complex process, so we'll see more of that in the future. And the bone cells, parathyroid hormone, will increase in resorption of calcium from bone. Skeletal muscle, epinephrine, conversion of glycogen to glucose. So glycogenolysis is increased, and glycogenesis will be attenuated at least. And in the intestine, the epinephrine will affect fluid secretion. And then the kidney, vasopressin, the resorption of water. And blood platelets, prostaglandin 1, the inhibition of the aggregation and secretion of the platelets. Okay, so this sounds like a list of the action of these hormones, the effect of these hormones, the metabolic response on the target tissues. But what we're saying is cyclic AMP in all these target tissues has diverse effects. And in only a few cases, like the skeletal muscle and the liver, for the same ligands, the hormones, we saw coupling to the same target systems. So this makes us think that there is a collective situation within a target cell where cyclic AMP is used as a second messenger system, depends upon the expression of other proteins downstream of the cyclic AMP to determine the metabolic response or the cellular response. So this is what we want to start thinking about, is same receptor coupling system, same second messenger system, excuse me, and then just same intermediate mechanisms. So how do you change the downstream effect? So this is what we're seeing here. Hormone 1 and hormone 2 both having an effect on the effector, the adenylate cyclase. We see the extracellular fluid, the cytosol here. And we know that these are G-coupled protein receptors, right? And the GDP gets kicked off when it's ligated, and the GTP goes on, and then it dissociates and can translocate within the membrane and bind to the effector. In this case, adenylate cyclase, ATP is converted to cyclic AMP. Downstream of that, the inactive protein kinase A for the A in cyclic AMP. And then that activates the protein kinase, and it triggers responses of the target cells, activates enzymes like the FSH and LH on steroid synthesis, stimulates cellular secretion, opens ion channels, right? So how do we couple cyclic AMP to the exocytotic system, and that's what we covered before. So catecholamines, ACTH, FSH, LH, glucagon, PTH, TSH, and calcitonin use this. And we know that in some cases there can be competing effects. If this was the lactotrope, then the prolactin releasing hormone, if it indeed it truly exists, would be using the G sub S probably to activate this. The D1 receptor uses the G sub I to turn it off, right? So you can have this releasing and inhibiting hormone signal transduction converging on the same second messenger system. You either increase the activity of the population of the enzymes or decrease it. Excuse me for just a moment. Okay, sorry about that. So now we can take a look at this figure. We see time on the x-axis and 1, 2, 3, 4 panels. These are 4 individual organisms, adult female rats that were overreactomized. It shows us the variation, but more so it shows us on a short time scale, minutes. This is just 2 hours, right? And the samples were collected every 5 minutes. And then the LH was measured by radioimmunoassay. So in the absence of hormone 3, we know that hormone 2 would go up. That's negative feedback. Remove hormone 3 and hormone 2 from the pituitary is going to shoot up sky high. But what it takes on is this pulsatile type of profile in the plasma. So remember, it's like the water level in the bathtub. The rate of the water going into the bathtub versus the rate of the water going down to the plug or the drain will determine the level in the water in the bathtub at any one point in time. So most likely the clearance rate of LH will have stabilized at maybe a new higher level, depending upon how long these animals have been overreactomized. But that would be thought to be rather constant. So what changes the level of the water, or LH, in the bathtub or the plasma is the cyclic release. So remember we talked about calcium oscillations and rhythmic exocytotic events, right? And so this is how we get these broad swings. We're going from 1200 nanograms per mil in the plasma of LH up to 2800, so more than doubling the plasma levels here just in 5 minutes. Right? So these are, and then essentially, so you're going to get just this pulse of release, and then for the next, I guess, 5, 10, 15, 20, 25, 30, 35 minutes, it's be, I guess, shut off to allow clearance, or gradually decreases, however you want to view it. If this is 35 minutes, what's the clearance rate of LH? If the clearance rate is 10 minutes, then we still have a gradual tapering of release, where release is more tightly coupled to clearance rate. Right? So if it's a 35-minute clearance rate, then what was released here took all this time to be eliminated. And then what's released here took all this time to be eliminated. So that means we're dumping it, and then we stop dumping at the pituitary, and we let it be cleared. But if clearance rate is higher, say 10 minutes, then you have to have a tapering rate of release, and the rate of clearance is going to now be high enough to exceed the rate of release to bring it down to a low level. And then the rate of release has to zip way up and then taper. So it all depends upon the clearance rate of LH. Is it more like 35 or 40 minutes, or is it 5 to 10 minutes? So this is the challenge for you to think about. How do we maintain at any one point in time the plasma level? So this is an overactomized adult female rat. So think of a perimenopausal or early menopausal human female. If you happened to draw the blood at this time, you'd get a low level, although it's still high, versus the normal. This might be a little more normal in here, like 400 to 800. But anyway, it still would be high, but if you happen to collect the sample at this interval, then right, so we go in and get our blood drawn. Is it fasting? Is it early morning, mid-morning, late morning? As long as it's fasting, who cares, right? They may not even care if you're fasting for the LH measurement. Typically, though, they measure FSH to test for menopause. But the idea is the clinician takes all of this into account. Regardless of when you would sample across this 2-hour time period, the average level should be higher than normal, right? So you're still going to get at least a 1200 and not an 800 or a 400. Now, who knows why this animal did not respond as robustly as this animal, and as well the others are a little bit lower. So there's individual variation, could be natural variation, could be this animal has an adrenal gland that kicks out hormones 3, and what we don't have here are the hormones 3 levels. Would they be absolutely 0 in each of these animals? Maybe in this animal there's a little bit of hormone 3, a little more of hormone 3 coming from the adrenal cortex, that would be attenuating this overall profile. But what's very interesting is this pulsatile release, right? Pulsatile release based upon the clearance rate, producing these ultradian rhythms. Ultradian, shorter than a day. Circadian rhythm is over a day, and of course the menstrual cycle is over on average 28 days. Okay, so we always have to think of our temporal scale. The hormone transport then, so we've got it secreted from the pituitary or the target hormone 3 from the organ 3, and we're thinking, and of course it's hormone 1 from gland 1, hormone 2 from gland 2, and hormone 3 from gland, hormone 1, 2, and 3 from gland 1, 2, and 3, sorry. It's the same principles apply. Rate of synthesis, rate of secretion, occupancy, as we're seeing here in the transport fluid. Is there something that holds it and protects it from degradation, right? And then, of course, there's the clearance. And all of those together contribute to an instant in time where you would see a certain concentration. Now, when we're taking into account these carrier proteins, then we're also, in the case of thyroid hormone, you may have heard that they'll measure free and bound thyroid. Same thing for testosterone in males. The amount of free testosterone that is not bound to the TBG, testosterone binding globulin. It's TEBG because it'll also carry estrogen. And of course, both males and females have both testosterone and estrogen in their plasma. Females have higher estrogen, depending upon their stage of reproductive status, and males obviously are going to have higher testosterone. Okay, so oxytocin and vasopressin have these carrier proteins. I'm not so certain they end up in the plasma, but they are in the vesicles, protecting these molecules on their way down the axon to the posterior pituitary. And then thyroxine and triiodothyronine, so T4 and T3, the thyrionine binding globulin, TBG. So the free is not bound to this. The free thyroid hormone is not bound to this in the plasma. The bound is bound to this in the plasma. And the concentration of the free hormone in the plasma is that which is readily available to diffuse down its concentration gradient into the target tissues, right? Into all tissues, and the target cells that's express the gene for the receptor will be the target cells in that target tissue. Okay. So the glucocorticoids and progesterone, a corticosteroid binding globulin. As I said, albumin is a globulin protein produced by the liver that also carries some of these, but it's not a specific one. These are more specific. Okay, so the ultimate state of clearance for nearly all of the hormones that we'd be thinking about is going to be in the urine. If they're amino acids based hormones, they might just be broken down ultimately into the amino acids. As we saw, the tensinogen becomes angiotensin 1, then angiotensin 2 as more and more amino acids are cleaved from it, and angiotensin 3 and angiotensin 4, and then maybe it's just completely broken down into single amino acids and released, and then recycled. But this is not always the case. The kidneys are working all the time, so the kidneys are going to have their chance to have the hormones filtered from the blood and not be transported back into the blood, or maybe not completely transported back into the blood. It all depends on how any hormone might be taken back up in the kidney. Of course, any of the steroid hormones, it's free diffusion based upon the presence of the carrier proteins favoring holding it in one location. Those large globulin proteins should not pass through the glomerulus, so they would not be in the filtrate. They would be in the filtered blood, passing past the glomerulus and to the peritubular capillaries, and be present to capture the steroids that are diffusing back in. Right? But some steroids are converted, like a 17-keto steroid, estrogens to estrones, and corticosteroids have many metabolites, prostaglandins converted to various fatty acids. These will not have plasma carriers, so they're most likely to stay in the urine, or at least produce a equal concentration in the urine as in the blood. That's why you can measure the concentration in the urine and have it reflect, be a biomarker for what's in the blood, right? So serotonin is the 5-HIAA, 5-hydroxyindolacetic acid, melatonin, 6-hydroxymelatonin, growth hormone, many metabolites. So growth hormone is a rather large peptide, polypeptide, actually protein, hormone, and it is glycosylated in many areas, so it's metabolized to many metabolites that can be found in the urine. So this is what we want to be thinking about. And then our final slide for this presentation, of course, is LH. Right? And so this is a fuzzy image, but anyway, it's a D-best one-step rapid test, an LH ovulation test. Right? So the test detects LH at 30 milli-international units per ml in the urine and above, which is high enough that gonadotropins in polycystic ovarian disease are not tested falsely positive as being ready to ovulate, and yet the LH ovulatory surge is detected. So luteinizing hormone LH is always present in human urine. It increases just before a woman's most fertile day of the month, and this LH increase triggers the ovulation. So we'll look at that more closely when we study the menstrual cycle. But this is and the pregnancy test is not going to look at LH, of course. It's human chorionic gonadotropin. So this reminds us that these hormones end up in the urine, and that's part of the way of getting water out of the bathtub. So we'll